Hello everyone and welcome to our lab on the root apical meristem. Today we're going to be taking a look at the internal structures in cross-section first of both dicots and monocots. Then we're going to be taking a look at a longitudinal section, meaning up and down, of a monocot root tip to see the different regions of that root tip as it grows down. And then last we'll take the most outward view and take a look at a root system as a whole. So let's begin by taking a look at our dicot root. So the first root, as outlined in the lab, is the root of the herbaceous plant, the ranunculus, or the buttercup. So here we have a couple of images of buttercups, trying to tie in some visible features that you might recognize. So we have a plant that is not woody, produces flowers. The flowers are in cymes, meaning that they have one region with the female parts, one region with the male parts, another region of the petals, and then behind that, a region of the sepals. And I wanted to include at least one picture of a local buttercup, Ranunculus repens, a very pretty little native that we found here in our county in Michigan. Now, when we're identifying the features of this root under the microscope, we're going to want to pay very close attention to the stele of the plant, what the central vascular cylinder looks like, where the epidermis is, where the cortex in that root is, where the endodermis is, based on the presence of a Casparian strip, and then the presence of primary xylem and phloem within the stele of the plant. So here we have that cross-section at 100x under the microscope. You'll notice that there is a vascular cylinder in the middle. It makes a quite distinctive cross shape. You'll see the stele here, the epidermis, this tissue layer on the outside. You'll notice that it is a very distinct color compared to all of the fleshy cortex, the parenchymal cortex, allowing you to differentiate between what is epidermis and what is cortex. And then let's take a closer look to see if we can identify the cell layer in particular that is our endodermis. So zooming in a bit on our slide, we see that this tissue, that is the cortex, bumps up against one very distinct layer of cells that look to have a thicker red-stained cell wall. That red-stained cell wall is the Casparian strip that we know from our lecture material forces water to be filtered symplastically rather than apoplastically. So if we're looking at the overall trajectory of how water goes from the outside of the root, it'll first bump into the epidermis, and because the epidermis is essentially made of very absorbent paper towels, paper towels are made from plant cell walls after all, it will flow apoplastically along the outside of these cells and flow very, very quickly through the cortex up until it bumps into our very distinct layer of the Casparian strip. Now the Casparian strip, again, forces it inside the cell to be filtered through our aquaporins. Once that water flows symplastically through the endodermis that has that Casparian strip, it'll then be absorbed into either the xylem, which are these larger red cells. You can see the difference between some of the primary xylem, the early made xylem, much smaller, versus some of our secondary xylem. Look how giant those vessels are. And same goes with our phloem, colored in teal here, or slightly blue, I suppose. These tissue layers that are going to be transporting our sugars. And so there you have it, the different pieces of our ranunculus, or buttercup root, visualized at 100x. Next up, we'll be labeling these same features, but in a different phylogenetic group, our monocots. taking a look at our smilax root. Well, not actually a root, here's our actual plant. This is a monocot, a member of the Liliales order, so it's not quite what you'd consider a typical grass, but it is kind of a distant cousin to the grasses. As a monocot, its cellular structure in the root is going to be very distinct from our buttercup. Monocots, characterized by their parallel veins, you see how these things, even though they come out of one point, they do generally tend to run fairly parallel to one another, are really characterized by that lack of secondary growth, the lack of ability to get truly woody with annular rings. So under the microscope, this structure is going to look quite distinct from our dicot. Now you are responsible for labeling all of the same features as we saw in our dicot root. So you should absolutely make note of the stele of this plant, a much, much larger stele here. In fact, the lab itself draws your attention to how much more of the plant root is taken up by the structures that transport things. You should also note the epidermis as well as an exodermis, a tissue layer on the outside of the epiderm providing a very thick, in this case, much more woody, even though it does not have wood growth, it is a much thicker outer layer on this greenbrier or smilax. Of course, then we also have our cortex, similarly to our buttercup root, and then an incredibly distinct endodermis. So here we have a quite distinct and incredibly thick Casparian strip. You'll notice that this is much more red stained than our dicot root. One tissue layer I actually forgot to point out on the dicot root is the pericycle. The pericycle being the layer of living cells directly underneath the endodermis, so inside of the root. Now this layer matters both in smilax as well as ranunculus because that's the tissue layer from which all of the root hairs are going to arise. So it's quite important. Now, in addition to our pericycle, we all, of course, have our layers of the xylem, the big vessels here. The protoxylem being the stuff outside of it, much smaller and spaced in between. Secondary xylem being our large vessels. And then, of course, our phloem. You'll notice smaller patches of phloem here. They're characterized by a purplish color, similar to what we saw in our dicot root, where the color change was very distinct from our xylem. And uniquely, we do have a living layer of cells on the inside here, inside of the metaxylem, a bit of our pith. So all in all, from the bird's eye view, it should be fairly apparent as to why the smilax root is very different from our dicot root. And that's what the lab is trying to get you to notice. Pay close attention to the stele, pay close attention to the discrepancy in the size of the anatomy of the stele, pay close attention to the difference in shape. You'll notice here on our smilax, the xylem is fairly circular, whereas when we look back at our ranunculus, the xylem forms a very distinct cross, a pattern that we see repeated over and over in lots of our monocot versus dicot comparisons. And to bring it full circle here, the pericycle that you see is this tissue layer of living cells right underneath our red-stained Casparian strip. So you should draw these structures both as they seem under the microscope, and then wrap up our comparison of the monocot and dicot root in cross-section by drawing some cartoons in the lab. So that way you have a simplified version to help you keep track of this long term. Well, hello everyone. So before we get to our microscopy of the root tip growth, I wanted to show you the model because I think it's quite neat. So looking vertically here, you'll notice the cross section of our buttercup root. You'll see the features that we just went over with our cross-shaped xylem, our phloem, our pericycle in yellow, and then our endodermis with the Casparian strip in brown. Cortex fully developed and epidermis fully developed. Then we turn sideways, and you'll notice different regions of growth here. So the xylem again is in pink as it differentiates, as it matures. Our phloem is in teal here, with some of our nuclei also stained in teal. On the base of the root, you'll notice a region that does not have cell staining. This here is our root cap. The end point of the root cap is actually dead at functional maturity. Because as I mentioned in lecture, this is a piece of the root that needs to help buffer the digging process. Because essentially the root is going to be digging with its face. So the very outer layers of cells are actually going to die in the process of the root digging deeper and deeper. So your direction of cell division is actually to keep pushing the root cap forward, whereas here the region of elongation is going to be pushing the root cap down. So you have these contrasting directions of growth that help a root grow in the direction that it needs to. So the root cap that's dead is furthest away. You'll have a layer of the living root cap. And then you have our different sections. The region of the meristem, where cells are dividing. You'll notice the cell size in all of this is much, much smaller in the region of the meristem, because as the region of the meristem proliferates, it then drifts up into our region of elongation, and the cells begin to lengthen. Their cell walls expand in the region of elongation. And then last but not least, up top, we have the region of differentiation. The region of differentiation is where you get that full maturity of the cell to become xylem, to become phloem. Remember, xylem dead at functional maturity, phloem still alive. The difference in those sieve tube cells versus companion cells. So you have this growth propelling the root cap down. And keep in mind that the mastermind of all of this are the hormones being secreted by our quiescent center. Somewhere here in the region of the meristem, there are those two cells that sit in the very middle of the root and secrete hormones to tell all of the cells around them to perform cell division. And then as you drift away from there, your distance from that quiescent center is going to establish when you enter the region of elongation, and your distance from the quiescent center and the region of elongation is going to determine when you enter the region of differentiation. I'll see you all on the microscope for how this looks in real life. All right, so now that we've had a quick rundown of what this looks like on a root model, let's take a look at what it looks like under a microscope, because the microscope and the model will, of course, be somewhat different. Here you can see at 100x a very, very distinct root cap. You can even see the part of the root cap that is characterized by the dead cells versus the living root cap. And then at 100x, we definitely see the region of meristem, where cells are very small and dividing. And we start to see the hints of the region of elongation. You'll notice that the blocks here in the region of elongation start to get much, much bigger than they are down in the meristem. And then, of course, when you look all the way up further on the root in the region of maturation, you'll see the fully elongated cells. So this monocot root, despite the fact that it has some differences with the dicot root in cross-section, will be very, very similar in longitudinal section. For the last section of the lab here, taking a look at some root systems, hopefully you've had a good time drawing the structures under the microscope, so I want to end by looking at things from a macro level scale, how these roots look like when you pull the plants out from the ground. Now, in the greenhouse, we'll go over the two characteristic root systems, the fibrous root system here, that we see very commonly in a lot of monocots. No primary axis, lots of spreading all over the place. And a taproot system, characterized much more by a thick root, that's the primary root, and much fewer secondary roots, or lateral roots. Taproots are often found in our dicots, like this dandelion here. Maybe you've pulled out a dandelion before, whereas our fibrous roots are found in our monocots. There's another type of root that you may see around this area, particularly if you are farming corn, and that is the system of prop roots or adventitious roots. These can be found over the ground or above the ground and can provide support to some of these plants. We'll round out the lab and finish today with a little demo in the greenhouse. I'll see you over there.
All right, for our last section of the lab, we want to take a look at the macro scale things, a little bit of root architecture overall. And again, just like in our lab, we were comparing monocots and dicot roots in cross-section. We're going to finish the lab by comparing monocot roots and dicot roots in terms of their overall architecture. So here we have a dehydrated palm, one I dried quite a long time ago, because its root system is very emblematic of this adventitious or distributed or fibrous root system. I prefer the term fibrous because this looks very much dispersed. You don't have a single axis. You have multiple roots with no one being more dominant than the other. This helps keep our monocots in the ground and absorbing a lot of water the moment it rains. So for those of you who've maybe pulled up grasses as a child, you'll remember that when you pull up the clump of grass, the grass on top is mirrored by the pattern of growth down below, where it's a very fibrous clump of grass, really allowing that plant to take advantage of not the deep reserves of water, but rather the very shallow reserves when it rains immediately. And we see that across all of our monocots, a much more fibrous root system. Now, when we pull up a very, very root bound, so this plant will have not as deep of an architecture as it should, but it will still have the pattern that we want to see.
Go.
All right, so here we have our Stapelia, a eudicot from Africa. Now you'll notice that it has kind of this fibrous root system in lateral roots because it's somewhat root bound, but you do see a much more distinct main root coming from it. Dicots are characterized by taproots, much deeper singular axes roots. Think of a carrot when you think of a taproot of a eudicot, a singular stem extending down into the soil. These taproots can go extremely deep. Rather than absorbing the surface water like we saw in our palm, eudicot taproots are trying to get into deeper aquifers. Some of the longest eudicot taproots can be multiple times longer than the growth of the plant above the ground. I'm talking 10 times more root length than shoot length above. So taproots go deep in a singular axis. They do have branching off, lots of root hairs and lateral roots growing again from the pericycle, but the architecture is different. One allows for a deeper growth, dicots, whereas one allows for a much more shallow and quick absorption. So have fun drawing those different architectures. I'd encourage you to see if you maybe have something around your home or around your house where you might be able to further investigate some of these root architectures. Finish up any drawings that you may have in the lab, and I will see you all next week.